Hey guys, this is Kendall Terry, and this is Cells Part 2. We've already discussed in the previous lecture cell theory, cell size, and what all cells have in common, and even some unique things about them. Now we're going to look at cell structures, and the cell structures we're going to look at primarily are the cell structures that you find in eukaryotic organisms, because it's going to be cell structures that are that have a membrane around them. So let's look at that. The first one is the endoplasmic reticulum. The endoplasmic reticulum is actually broken into two types, the rough ER and the smooth ER. The first thing that's the difference, and you'll see here with the rough ER, is the presence of ribosomes. That's actually what give the rough ER their name. Under an electron microscope, the rough ER have ribosomes on the edges, and it makes it appear like little dots on the edges or rough looking, whereas the smooth ER do not. But that's not the only difference, and you can see here on the screen there's much more to it. The rough ER is responsible for making proteins set to leave the cell. Now this could be proteins that are going to be used, maybe like an enzyme or a hormone. It could be a protein that's going to be used on the cell membrane itself and it's going to stick off the cell membrane or through the cell membrane. Those are going to be made through the rough ER. As opposed to the smooth ER that's used, it makes stuff, it has enzymes in it, that catalyze the synthesis of carbs and lipids. It also stores calcium ions that the cell uses in signaling, and those calcium ions are actually extremely important. But if they were just floating around inside the cell, it would be it would cause different reactions to take place that you don't want to have happen. So the smooth ER in this case acts kind of like a storage compartment for these calcium ions, and then when they're needed, they will be signaled to be released. And then they detoxify on foreign substances. So as your body, as you eat things or as your body is breaking stuff down, it may make something that could be harmful to it. The smooth ER is one of the systems that the cell will use to detoxify that. You see a lot of smooth ER and stuff like liver cells, for instance, and it helps detoxify alcohol. That's one of the places that it will actually function. Another is the Golgi Apparatus. So the Golgi Apparatus, when you look at a cell, looks similar to the smooth ER. So you have to be careful not get those confused and make sure when we're doing some stuff in class that you pay attention to the difference. But the Golgi Apparatus is a collection and packaging center. Many people think of it kind of like a post office. They're gonna process stuff and then send it out the back. It actually has a front and a back. There's distinct parts of it. Stuff comes in on one side, it leaves on the other. It doesn't ever go the other direction. And it's also involved in the synthesis of the cell wall in plant cells. This is a function that it does not have in animal cells, but in plant cells, as mitosis is finishing, cytokinesis is finishing, the cell membrane, the cell is needing to be split in two, you actually get a cell plate formed in between those two cells, and it'll be an area that's surrounded by Golgi apparatus for a while until that cell plate develops into a true cell wall. The next type of cell structure is the lysosome. Lysosomes are kind of like the garbage men of the cell. They have their digestive vesicles that arise from the Golgi apparatus, and then they go around the cell and they're looking for anything in the cell that needs to be broken down, and they engulf it, break it down, and then get rid of the byproducts. For instance, an issue here is Tay-Sachs disease, one that we'll talk about more in class, even in other chapters like genetics and things like that, is a loss of the function of a single lysosome enzyme, and it can no longer remove fat in the brain, so you end up with literally like a fatty brain type situation is what is kind of the way to describe it, but that causes then your nerves not to be able to function right and eventually loss of life. So when we look at cell structures, there's a category of two cell structures that we're going to pay attention to here, and those are cellular energy organelles. The two are mitochondria and chloroplast. They're both surrounded by a double membrane. They contain their own DNA, and they make their own proteins. And though this is unique to these two organelles, when you compare these organelles to others, these are the only two that contain their own DNA and that make their own proteins. Now, they don't make all of the proteins they need to function, but they do make a lot of the proteins they need to function. The mitochondria is about the size of bacteria. The animal cell is much larger than a bacterial cell, and the mitochondria in the animal cell and plant cell both are about the size of bacteria. They have a smooth outer membrane, and the inner membrane actually folds back and forth. And when you look at it, it looks like kind of like a sausage with a worm inside of it as that smooth inner membrane, or that inner membrane, folds back and forth. And this creates these turns that we call cristae. And you can see that right here on the screen. This makes many of the enzymes needed in cellular respiration, because that's what mitochondria does. It's the point of the cell that does cellular respiration, that breaks down sugars and other molecules to make ATP energy. ATP energy is what causes the cell to be able to function. So without mitochondria, a eukaryotic cell would have to get its energy source from something else, and it probably wouldn't survive. So it's very important in survival. And this divides in two during cell division. A lot of the organelles inside of the cell actually just dissolve, or just kind of get split apart. But in mitochondria, they actually go through a division process very similar to bacteria in binary fission. In the chloroplasts, these are the food manufacturing centers for the cell. You see these in plant cells. They contain, that shouldn't be chloroplast, sorry, that should be chlorophyll, not chloroplast. The chloroplast contains chlorophyll, and they also have compartments of stacked membranes called grana, and they literally look like little discs stacked one on top of another. If you think about maybe an Oreo cookie, and you stack a bunch of Oreo cookies on top of each other, that's kind of what looks like is happening. Each grana contains several dozen disc-shaped structures called thylakoids, and that's what, each cookie would be, a thylakoid. So you've got, and in these thylakoids then is where that chlorophyll is located, and that's where photosynthesis takes place. That's where the sugar molecule is made, and that sugar molecule is really the energy source that we need as animals, to make all the ATPs that we make. Very important. So the final area right here, if you're getting lost, this is cell structures, and here is cytoskeleton. This is the final thing that we're going to look at. There's three types of cytoskeleton. This is probably a new thing for you that are in Bio 2 that you haven't touched on much. This is actin filaments, cellular movement, contraction, crawling, pinching during cell division, all come from cytoskeleton molecules called actin filaments. And they're really cool. They do help some pretty cool stuff, like the contraction of your muscles, crawling. You don't think of your cells crawling, but those immune system cells, when they come to like a cut in the skin, will literally get to the edge and then crawl out into the surrounding areas there to look for any invaders and pathogens and stuff like that, to take care of it. Microtubules, these are for movement of chromosomes, organization of to the cytoplasm, stuff like that. So whenever you look at mitosis, for instance, going on, and you'll see that the chromosomes get lined up in the middle and you have these spindle fibers, those are microtubules that connect those chromosomes and move them around and then pull them to opposite sides and stuff like that. And then you have intermediate filaments. These give the cell mechanical strength. These intermediate filaments sometimes will be between cells and they'll be linked together. Sometimes they'll go from one cell into another cell, and even attach to parts of that other cell, and that causes that anchoring to take place. You'll see intermediate filaments even between cells in your small intestine to create these really tight junctions, so that food molecules have to go into a cell to get into your bloodstream, and they can't just pass between a cell. You don't want food molecules just being able to get into your bloodstream. So they have to go through that selectively permeable membrane to get into that cell, to really get into your blood system then, so that your body can use it. The hope being that whatever gets through that cell is important to you.